Professor Amitava Adhikary is a Research Associate Professor, Department of Chemistry at Oakland University. His research area includes elucidation of the mechanistic pathways involved in direct interaction of radiation with DNA, formation of site-specific amino radical.
Good afternoon, organizers. First of all, I extend my heartfelt thanks to Professor Gopinath for inviting me and giving me the opportunity to present the results that is related to ion beam radiation therapy, the fundamental radiation chemical mechanisms for this. So here we go. Before we began, actually, around 2002-2003, the idea was that excitations in causing DNA damage, the early views were for the low LET radiation like gamma, excitations had been thought to not cause DNA damage. Now, as we all know, when radiation interacts with DNA, because of the ionization events, the electron loss leads to DNA cation radicals, that is the DNA dot plus, as you can see, which are also called the holes, and these DNA cation radicals, they cause DNA damage by oxidative pathway, and the excess electrons, they are produced because of the ionizations. They are captured by the electron affinic sites in the DNA molecule, producing DNA anion radicals, and these DNA anion radicals lead to DNA damage by reductive pathway. And the simple model, of course, I'll show you in the immediate slides, that includes the contributions of excitations and low energy electrons. Now, in the next slide, we show the formation of the DNA damages by oxidative pathway, and I am very happy to inform that over the last 20 years, our lab has been able to identify all the radicals involved, and we are also characterizing the molecular, diamagnetic molecular products, stable molecular products, with the help of NIST, the National Institute of Standards and Technologies, with the help of Dr. Miral Dizdaroglu. Now, what does this slide mean? That as we all know, the ionization of the DNA is a random process. So all the bases and the sugar phosphate backbone, they will all be ionized, and the cation radicals thus formed will be localized to the most electropositive base, guanine. And as you can see, when guanine cation radical is formed, guanine cation radical being either a Lewis acid or a Bronsted acid. Now, what we know about the acids, they lose protons, right? So therefore, the guanine cation radical, just like an acid, will lose a proton, form the neutral radical or its conjugate base. That is one reaction called prototropic equilibrium. The guanine cation radical also undergo nucleophilic attack of water, producing the GOH radical, which can, by one electron oxidation, leads to one of the most important molecule, 8-oxoguanine or 8-oxo-2'-deoxyguanosine, and the same radical by one electron reduction producing the PAPI, formamidopyridine guanine damage. Now, the guanine cation radicals, if they are very close to an excitation event, in fact, we have shown that if the guanine cation radical can be excited by these photoflood lamps, they can lead to form the sugar radicals. And those backbone cation radicals that escape the backbone to base whole transfer process, they can also undergo deprotonation, producing sugar radicals, which are the precursor of strand breaks or the base release. Now the same thing, similar mechanisms happen for the DNA anion radicals. As I mentioned, because of the ionization events and the electron capture, these events are random, so all the bases and the sugar phosphate backbone will capture electrons, and through the electron transfer process, the electrons will be captured by the most electron affinic bases, thymine and cytosine, producing the anion radicals. And just like I told you in my previous slides that the cation radicals are Lewis acid or Bronsted acid, they lose protons. Similarly, the anion radicals are Lewis base or Bronsted base, they accept protons. So thereby, by accepting protons, then by undergoing another reaction and another protonation, they will lead to the dihydrothymine, dihydrocytosine, dihydrouracil, and the dihydrouracil comes from the deamination of the dihydrocytosine. And some studies, they have shown that this particular deamination process possibly is linked to oncogene activation. The most important process here, when the electron attaches to the sugar phosphate backbone and that undergoes a particular reaction calling dissociative electron attachment. Now let me show you this very clearly. Now you see the electron is attaching to the sugar phosphate, forming the corresponding anion radical, and this by either carbon-oxygen bond for breakage, which is the most favorable pathway, that is 95%, forms the diphos radical. That means sugar phosphate bond is already broken, which is a strand break, and then at the same time it is forming a radical site in the sugar moiety. That means one event is leading to two different type of damages, which in the terms of biochemistry we call formation of clustered lesion. So this particular pathway is very important. Now, with all this knowledge, I'm coming to the ion beam. So in the ion beam radiation, what we find is that the total yield of the stabilized radical is lower than the gamma radiation, which is the low LET, but the ion beam radiation leads to the higher relative amount of the backbone radical formation. And we have two hypotheses to account for this. One is the excited state process, which I already mentioned, that the cation radicals or the anion radicals in the excited state form the sugar radicals, number one. And then number two is the involvement of the low energy electron, which I already showed you that the low energy electron leads to the formation of not only strand break, but also another radical site leading to the formation of cluster damage. So now, in terms of physics, what do we understand? You see, here I am showing two different types of radiation. One is the low LET radiation, which is an electron track, and then the other is the alpha particle track, which is the high energy, high LET radiation. Now the low LET radiation, as you see, because of its track structure involved a lot of spurs, so therefore we have lot of individual damages. But on the other hand, when we see the alpha particle track, because of the high LET, that means a large number of energy being deposited per unit track, so therefore the formation of the cluster damage is much more in high LET radiation than in low LET radiation. And this actually we have proven in our lab with DNA samples like this. our colleague Dr. David Baker came up with this idea of DNA sample preparation so that we can not only see the Bragg peak due to high LET radiation, but on the radical level, but also when we warm this irradiated sample to room temperature and slice it a number of pieces in order to identify the products in the lab of Dr. Miral Dizdaroglu, then we can compare or we can translate in the same sample the radicals converted to products. And you see, this is an example. I'm sorry. So, let me first show you this and then I go back. So you see, this is a classical example of a tandem lesion, 5' 8 cyclopurine 2-deoxynucleoside. This is a tandem lesion that is difficult to repair, and as you can see, not only we have shown that these lesions are formed in ion beam irradiated DNA, but we have also been able to identify which conformation of this lesion are formed in solid DNA samples, and the conformation is R. On the other hand, in aqueous solution, the S conformation is favored. And when we wanted to translate these results in cells, we found that the Professor Al Fornes's lab at Georgetown University found predominance of R 5' 8 cyclo 2-deoxyguanosine in ion beam irradiated cells from mouse. And we have measured more or less all the different types of base damage products, and in all cases what we found is that the maximum amount of the damage happened just before the Bragg peak. Now why it is that? You see, in the ion beam radiation or in the high LET radiation case, which, let me go back a little bit further. Yeah, so as you see, because of the track structure of the high LET radiation, lot of energy is deposited in the core. So what will happen? The number of recombination will be maximum. And so in the core, the amount of the sugar radical formation will be the maximum. And that's what we find. And then in collaboration with Johns Hopkins University, I am doing Gen4 DNA calculations, and what we are seeing is from this calculation, the result is that for the electrons being low energy radiation, the cluster size is less, the amount of DNA damage is high, but on the case of alpha particle, the cluster size is much more. So you see, the theory and experiments, they are going hand together. Now what is the conclusion of these studies? Now this is a unique study that is a combination of the electron spin resonance spectroscopy and the tandem mass spectrometry application, and to find the DNA radicals and its products in the same sample. And what is most important is the maximum amount of the product is found just before the Bragg peak. And therefore, this work actually provides a much better understanding of the mechanisms of the radiation damage along the ion beam track in terms of the formation of the DNA radicals and in formation of the stable diamagnetic products. And so I will be organizing the 16th International Workshop of Radiation Damage to DNA. And here are our group. and my collaborators, and I have lots of students, and one of my joint supervised student, Mr. Jishnu Narayanan from IIT Mumbai is present here. And I'm grateful to my funders, and thank you very much, and I welcome questions.